دكتورة تفضل.
Good morning. My name is Dr. Iman bin Dayed, an assistant professor in the Community Health Science Department in the Nutrition Program. And it's my privilege and pleasure on behalf of the Community Health Science Department to welcome you here today. Today, we will have Ms. Hadil Bdawi master thesis discussion. We are honored and delighted to have our examiners, Dr. Dara Disi from the Community Health Sciences Department in the College of Applied Medical Sciences, and our external examiner, Professor Afaf Al-Ansari, a professor in biochemistry, a senior scientist in the Central Laboratory, Female Center for Medical and Scientific Studies at King Saud University. Ms. Hadil's presentation is titled Vitamin D Status in Children with Cystic Fibrosis. It is associated with gut microbiota and pulmonary function. Before I hand over to Ms. Hadil, our master students who will present today, I want to say once more on behalf of the Examiners Committee, welcome. It's wonderful to see so many of you here today. Please welcome with me Ms. Hadil.
Good morning, everyone. This is Hadil Bdawi. Today I will present my thesis that was supervised by Dr. Iman bin Dayed. So the title of my thesis is Vitamin D Status in Children with Cystic Fibrosis. Is it associated with gut microbiome and pulmonary function? So in this presentation, I'm going to go through introduction, the aim and the objectives of the study, the method we used, then the main result, and discussing the main findings. So I'll start with an overview of cystic fibrosis. Cystic fibrosis is an autosomal recessive disorder. It's caused by the mutation of the cystic fibrosis transmembrane conductance regulator gene. So the main function of this gene is the transportation of chloride, bicarbonate, and sodium and water across the cell membrane. Whenever there is a mutation, like in cystic fibrosis, there will be a disrupt of the transportation. And this disruption of transportation is what shapes the manifestation of cystic fibrosis. The manifestation include chronic lung inflammation, repeated infection, pancreatic insufficiency, and gastrointestinal abnormalities, and malnutrition, and fat malabsorption. And here I would like to focus on the fat malabsorption, as it's the main cause of vitamin D deficiency that's often seen in individuals with cystic fibrosis. This fat malabsorption is caused by pancreatic insufficiency, and it leads to vitamin D deficiency. There are other causes of vitamin D deficiency in this population, which is the lower expression of vitamin D binding protein that's caused by the corticosteroid medication used for treating inflammation. Also, these individuals often have repeated antibiotic use, and some antibiotics have the side effect of photosensitivity. So this results in inadequate sun exposure. Also, the nature of disease may lead to restricted mobility, and this also leads to inadequate sun exposure. All of these factors result in vitamin D deficiency. And maintaining a normal level of vitamin D is especially important in cystic fibrosis because it can have an impact on respiratory outcome. So 25-hydroxyvitamin D can or have the capability of increasing antimicrobial peptide, and this will resist the Pseudomonas aeruginosa, which is the pathogen that often individuals with cystic fibrosis presented with. This pathogen affects the airway. So vitamin D have the ability to reduce the colonization of airways. Now moving to gut microbiome. Now, in any healthy host, the gastrointestinal tract is inhabited with a large number of microbial communities. The most dominant are the Lactobacilli, the Enterobacteriaceae, and Bacteroideaceae. And the importance of the gut microbiome. It's related to its role in mucosal regulation and intestinal immunity. And there are three pathways where gut microbiome could modulate the intestinal immunity. First, through defending the gut from the pathogen, through either competing for nutrient or secreting antimicrobial peptide. The second pathway is through the development of regulatory T cell that produce anti-inflammatory cytokines, such as interleukin 10. The third pathway is through promoting epithelial barrier, either by maintaining a thicker mucus or producing metabolites such as short-chain fatty acids. Now, individuals with cystic fibrosis are expected to have alteration in gut microbiome. This is because of repeated use of antibiotic, because they have changes in their gastrointestine, the mucus and the pH level, and also due to fat malabsorption, due to impaired immunity. All of these alter the composition of gut microbiome. And this alteration displays as lower diversity of Bacteroides and Firmicutes and higher diversity of Enterobacteriaceae, especially E. coli. There are several pathways where vitamin D could modulate gut microbiome. And these pathways include the reducing of intestinal inflammation, decreasing the permeability of gastrointestine, which happen through increasing the barrier function, and also stimulating the antimicrobial peptide in the intestine. Vitamin D is also important not only for the modulation of gut microbiome, but to its role in the immune system through suppression of helper T cell that produce pro-inflammatory cytokines and modulating the inflammatory cytokine that's produced by regulatory T cells. So this is an indirect way where vitamin D could modulate gut microbiome. Now, there are several interactions between the cystic fibrosis manifestation and gut microbiome. So with the dysbiosis of gut microbiome that's caused by the differences or changes in GI and mucus and pH, the dysbiosis or alteration or the gut microbiome imbalance leads to the production of pro-inflammatory mediators, and these initiate low-grade inflammation. This might result in systematic inflammation and eventually chronic lung inflammatory illness. Also, the lower secretion of chloride and higher uptake of water that's caused by the mutation of the CFTR gene leads to dehydration and increase the acidity of the intestine. This also compromises the absorption of nutrients. The impairment of mucus production and transport would create a suitable environment in the for the bacterial overgrowth. Now, the significance of our study is that we hope that determining the association between vitamin D, pulmonary function, and gut microbiome, cystic fibrosis, would help the healthcare professional to determine the optimal level of vitamin D and reach that with supplementary vitamin D, and also to seek different approaches to modify gut microbiome for better cystic fibrosis outcomes. Our aim is to assess vitamin D level in cystic fibrosis and non-cystic fibrosis controls. Then we measure fecal abundance of 5 of the most relevant bacterial species. Then we would investigate the association to vitamin D status and pulmonary function. So moving to our objectives. First, we want to determine serum vitamin D level in cystic fibrosis and non-cystic fibrosis control groups. Then we want to determine pulmonary function measured by FEV1, FVC, and FEV1 ratio using a spirometry in cystic fibrosis group. Then we would determine the gut level of 5 selected bacteria: Bacteroidetes, Bifidobacteria, Firmicutes, Lactobacillus, E. coli, and the total bacteria in cystic fibrosis group. Then we would determine vitamin D-related dietary factor assessed by a validated food frequency questionnaire obtained from a Musharraf study in the CF group. Then we would relate that to serum level and recommended dietary allowance. After that, we would like to determine the relationship between all of these variables. Now, moving to methods. So our study design was cross-sectional, and we've recruited children from King Faisal Specialist Hospital between July and December 2019. The cystic fibrosis had 35 children. We've measured anthropometric, serum vitamin D, dietary vitamin D intake, pulmonary function tests, and gut microbiome composition using real-time PCR. For the control group, we had 24 children where we measured anthropometric and serum vitamin D. Now I would like to go into details with all of these measurements. So the anthropometric measurement, we've calculated BMI Z-score and BMI percentile using the Center of Disease Control growth chart, the CDC growth chart. And their cutoff for BMI for age were underweight if below 5th percentile, healthy weight between 5th and 85th percentile, overweight between 85th and 95th percentile, and obese for over than 95th percentile. For the total 25-hydroxyvitamin D status, it was measured using Lexis vitamin D total 2 assay in the laboratory of King Faisal Specialist Hospital. And we've used a cutoff point for vitamin D deficiency and insufficiency from the Institute of Medicine. So their cutoff is that vitamin D deficiency is considered when the vitamin D level is below 30 nanomole. Vitamin D deficiency is when the levels between 30 and 50 nanomole per liter, and the optimal vitamin D level is over 50 nanomole. The dietary vitamin D intake was assessed using validated food frequency questionnaire. It was specific for calcium and vitamin D. We've entered the data in a Saudi database obtained from Al-Khalaf study. And we've measured the amount of vitamin D using the USDA food composition database. So moving to pulmonary function, the pulmonary function tests were performed by a respiratory therapy technician using spirometry at the pulmonary function test lab in the King Faisal Specialist Hospital. And the cutoff point were used for the severity of lung disease or airway obstruction were mild for FEV1 over 80%, moderate for FEV1 between 50 and 80%, and severe for FEV1 below 50%. And moving to fecal microbiota analysis. So starting with DNA extraction. First, we kept the fresh stool samples in sterilized container at 4 degrees We transported the samples within maximum of 3 hours. We obtained 2 aliquots weighing 250 milligram, and we stored them at -80 until DNA extraction. So we performed the DNA extraction on 1 aliquot using a commercially available kit, and we've maintained the other aliquot for repetition. Then we measured the concentration and the purity using nanodrop spectrophotometer. The extracted DNA was adjusted to the concentration of 10 nanogram per microliter, and then it was stored at 20 until real-time PCR. Then we had to build up a standard curve using a 10-fold serial dilution of DNA extracted from pure culture with known concentration that range from 100 millions to 100 colony-forming unit per ml for every reference bacteria. Then we've done the real-time PCR reaction run and plotted the CT value against the log concentration to create a linear regression. So the real-time PCR we performed was in 50 microliter reaction. We have done it in triplicate, 3 repetitions. We've used cyber green PCR master mix, forward and reserve primer, and 10 microliter of DNA. The initial cycle was 95 degrees for 15 minutes, then followed by 40 cycles of denaturation at 94 for 15 seconds, annealing for 30 seconds, then extension at 72 for 40 seconds. For the statistical analysis, first we've used a Shapiro-Wilk test to check normality, independent sample t-test to compare between cystic fibrosis and non-cystic fibrosis group. Then we've used one-way ANOVA to assess the differences in lung function and gut microbiome composition across the different group of vitamin D. We've also used Pearson's correlation coefficient to analyze the correlation between vitamin D status, pulmonary function, and gut microbiome composition. So moving to results. First, the demographic characteristics. So as you can see in table 1, the mean age for cystic fibrosis children was 8.7, and for non-cystic fibrosis was 9. There was no significant difference in terms of age or gender. However, we found a significant difference in region. Most of cystic fibrosis children were living in Eastern region, about 54% compared to 8% from the control group. Moving to anthropometric characteristics, the cystic fibrosis group had a significantly lower BMI score when compared to non-cystic fibrosis group. Their average BMI percentile was below the 15th, while the control group had an average of minus 0.23, which is below the 15th percentile on the CDC growth chart. Now moving to serum vitamin D level. So cystic fibrosis had significantly lower serum vitamin D, as you can see in figure 2. And their intake of vitamin D was 60% of the recommended dietary allowance for vitamin D and 69.8% for calcium. Moving to pulmonary function testing. So 43 of our sample had mild airway obstruction, while 26% had moderate, and only 31% had severe airway obstruction. We found a significant positive correlation between BMI Z-score and each one of pulmonary function tests. As you can see in figure 3, a positive correlation between BMI Z-score and FEV1 test, another positive correlation with FVC, and the third one, a correlation with a FEF test. So BMI Z-score was positively correlated with all the pulmonary function tests we've measured in the study. Now moving to gut microbiome composition. As you can see in figure 4, the most dominant bacteria was E. coli, and that was followed by Bacteroidetes. And the least abundant was Firmicutes and Bifidobacteria. We found no significant correlation between BMI Z-score and gut microbiome composition, both when measured as continuous or categorical. Now moving to vitamin D and general characteristics. We found no significant difference in terms of age, gender, region, or BMI among the different groups of vitamin D status. When we assess the association between vitamin D and pulmonary function, we found that pulmonary function was similar across the different groups of vitamin D. Except for borderline significance in the FVC ratio between the insufficiency and deficiency. But when we run a Pearson coefficient correlation test, we didn't detect any significant correlation between vitamin D status or vitamin D dietary and supplement intake and pulmonary function markers. So when we assessed vitamin D status and gut microbiome, we found no significant difference in Bifidobacteria, Lactobacillus, E. coli, Firmicutes to Bacteroidetes ratio across different vitamin D group. However, children with cystic fibrosis who had the optimal serum vitamin D level had significantly higher Bacteroidetes, Firmicutes, and total bacteria. This is represented in figure 6. The first one was Bacteroidetes and serum vitamin D. There was a significant difference between optimal and insufficiency group. The second one in Firmicutes, significant difference between optimal and deficiency group. And for the total bacteria, also significant difference between optimal and deficiency group. Also, this association was confirmed using Pearson coefficient correlation. There was a positive correlation between vitamin D status and Bacteroidetes, total bacteria, and Firmicutes, as you can see in figure 7. So this association was also confirmed with a positive correlation. So when we assessed the association between gut microbiome and pulmonary function, we found that a Firmicutes level was significantly higher in the mild airway obstruction group when compared to moderate group, while the rest of the selected bacteria did not differ across the different airway obstruction severity group. As you can see in figure 8. And also this association was confirmed with the correlation between Firmicutes to Bacteroidetes ratio and pulmonary function measured by FEV1 and FVC. It was a borderline significant. The p-value was 0.051, 0.052. As you can see in figure 9. We've also used a stepwise multiple linear regression to predict the severity of lung disease. And we had only 2 significant predictors. Age was a negative predictor of the severity of airway obstruction, and BMI Z-score was a positive predictor of the severity of airway obstruction. Now we're going to discuss the main findings of this study. So first, this study presents a novel assessment of the association between vitamin D, pulmonary function, and gut microbiome in children with cystic fibrosis in Saudi Arabia. And our primary objective was to assess serum vitamin D in cystic fibrosis in comparison to non-cystic fibrosis children. And this study showed a significantly lower serum vitamin D level in cystic fibrosis children. And this finding supports the previous observations that suggest a lower level of vitamin D in cystic fibrosis children. As for the BMI, we've showed a significantly lower BMI and BMI for cystic fibrosis children. And this could be explained by the maldigestion and malabsorption that's often seen in cystic fibrosis children. Also, their increased energy expenditure due to the nature of the disease, and the decreased energy intake that result from the GI symptoms they usually have. Then the regression model showed BMI Z-score to be a positive predictor of the severity of airway obstruction. And this was also reported in a previous study. The reason could be that the lower BMI in this group usually means lower lean body mass. And this results in weakness of pulmonary muscles and eventually leads to decreased pulmonary function. So in the study, we did not detect any association between vitamin D and pulmonary function. And the possible explanation for this might be that the majority of our sample had mild to moderate airway obstruction. 69% of our sample had mild to moderate. So there was no severe or pulmonary exacerbations. And in the literature, a few studies could not detect any significant difference or insignificant association between vitamin D and pulmonary function, but they did, however, find a significant association with pulmonary exacerbation. Now, our study showed that E. coli was the most dominant among the selected species. And this observation suggests there is a positive correlation between the abundance of E. coli and GI inflammation, indicated by calprotectin in addition to fat malabsorption that was reported in a previous study. Another study also showed that 25% of cystic fibrosis patients had E. coli lung colonization. Another study showed that this colonization is positively correlated with pulmonary function. But the link between gut and lung E. coli colonization is still unclear and needs to be determined in future studies. In the relationship between gut microbiome and vitamin D, we found that bacteroidetes, firmicutes, and total bacteria were significantly higher in the optimal vitamin D group and positively correlated with vitamin D. And these results were also reported in a previous study where bacteroidea which is a bacterial class where bacteroidetes belong to, was also higher in the optimal vitamin D group. We've also found a negative correlation between vitamin D status and E. coli. And this previous study also shown a significant association between the lower vitamin D level and the higher abundance of gamma proteobacteria, which is the class of E. coli bacteria. We've also found that higher firmicutes was shown in children with mild airway obstruction and compared to moderate. And this also was reported in a previous study where firmicutes were significantly higher in children with mild lung disease. This could be explained by the role of firmicutes in producing a butyrate that downregulates the pro-inflammatory factors such as interleukin-6 and interleukin-12. We've also found the ratio of firmicutes to bacteroidetes to be positively correlated with pulmonary function. And this could be related to the unusual reduction in the firmicutes to in relation to bacteroidetes that we found in cystic fibrosis population. So our study have some strengths and limitation. So the main limitation of the study is the absence of gut microbiome composition assessment in the non-cystic fibrosis children. In addition to the lack of intestinal inflammation assessment and the genotype data, we've also excluded unstable cystic fibrosis children. So we couldn't detect the relationship or the effect of pulmonary exacerbation. Also, the food frequency questionnaire we used was specific for vitamin D and calcium. So we could not assess the association with other micro and macronutrients. Also, this study has a cross-sectional design, so this neglects any potential prospective effect. However, we have some strength for this study. First, it is a nutritional inclusive study. We've considered nutritional status factors such as BMI, BMI Z-score, and nutritional intake of vitamin D and calcium. It's the first study to assess gut microbiome composition in cystic fibrosis children in Saudi Arabia. And we've collected the blood and stool samples the same day the spirometry was performed. Also, we've eliminated the use of antibiotics for at least 2 weeks to minimize their effect on gut microbiome. Also, the real-time PCR reaction that we performed was in 50 microliter, and we've took an average of 3 replicates, and there has been a minimal variation between them. So, in conclusion, our study showed that cystic fibrosis children have significantly lower serum vitamin D compared to non-cystic fibrosis children. And yet the study has been unable to demonstrate the association between vitamin D and pulmonary function. We found that gut microbiota composition was significantly different across different serum vitamin D groups. We've also found firmicutes level to be higher in children with mild airflow obstruction compared to moderate obstruction. So our study is strengthening the idea of modifying gut microbiome to get better outcome in serum vitamin D and respiratory outcome in cystic fibrosis. We recommend further study that used next generation sequencing technique that followed by real-time PCR and assessing gut microbiome to define other species that might be related to cystic fibrosis. Thank you all for listening. Do you have any questions or comments?
Thank you, Hadil. as we are bringing the opening discussion to a close, I want to thank everyone for attendance today and hope that the presentation has given you all a much clearer understanding of the relationship between vitamin D and gut microbiota in the cystic fibrosis pediatrics. We would resume with a discussion in a closed session now. So have a great day, everyone, and I hope to see you again soon.
اوكي دكتور اعطونا بس دقيقة ونقفل اللايف. لحظة بس لو سمحت.